Okay, so hi everyone. So
Nice.
let's get started with this paper, which I found really fascinating, and is scaling large language models for next generation single cell analysis. So before we dive into the paper, this is the structure that I want for the paper, and I wanted to have some type of baseline so everyone can understand the what the paper is about. So the first 5 minutes will be just fundamentals on biology specific to the paper, and I going to sacrifice a bit of accuracy for explainability, so we can kind of have that going. Then we go to the introductions, questions, result questions, and then methods question and the final conclusion. But feel free to ask any question at any point or any suggestion or anything that you want to share.
Nice.
So yeah, so a bit of context so we can kind of try to understand what's the importance of this type of paper and this type of research. And in the UK, approximately 1 in 2 people will get cancer at some point of their lifespan, which is 50% of us. So the number is pretty high, and everyone has relatives that have been through cancer or even ourselves in our lifespan. But if this doesn't feel that close to you, this might, and this is a father sharing the picture of her daughter, just to raise awareness and to also try to encourage people to research in cancer. So it's an important topic, and this young girl died from neuroblastoma, which would make sense when we develop more the research paper. Okay, so a bit of background. We are a living organism and we are composed by cells. Inside our cell, we have a nucleus, and inside the nucleus, we have chromosomes, and the chromosomes has certain regions that are our genes, and our genes are made of as the fingerprint of life in Earth, which is the DNA. And the collection of DNA is what makes the, the genes. So this is a lot of data, and we do have this amount of data available, but we don't have the ability to read all the data or to make sense all the data that we have are at disposal. It's like having a huge library without a librarian to make sense of everything that we have available. So that's what this research is important. And one thing that I want to emphasize is how important and how critical is the order for biology. So order matters a lot, and this is nucleotides, and those nucleotides, the composed of several nucleotides, is what it makes amino acids, and amino acids became proteins, and those proteins is what we have in all our body. And this is a gene, and the specific gene sections, they have in a specific order, and that order allows to read through or express our genes. So we even have regions to tell the specific proteins of body where they need to start to reading our genes, so we can develop everything that we have. But what I want to emphasize is how critical is the order for this, and that would make sense when we go to the fundamentals on what they did. So also one thing that I wanted to show you guys is that we have different cells in our body, but we have a common ancestor, and that common ancestor is basically a cell that can be anything that it develops. But as it starts to develop, it will activate and deactivate genes. So it's a matter of activation on deactivation into a spectrum. And that spectrum is what it will make different cells in our body that has different type of functions. And whenever you have this type of cell, for instance, that has some activated genes or more active genes than this one in certain regions. So that's important for what they did. So basically, this paper, what they wanted to accomplish is to have a language so you can pass to the LLMs and from the LLMs to try to make sense of the data without having to change the whole architecture of LLMs and also leverage their potential and making better answers when the developments of LLMs start to grow. So essentially you have a like this is a natural language where you have the genes and you ask, okay, what's the cell of these genes or where it belongs, and it will provide an output. And also it allowed to test you some perturbation response. So for instance, okay, I have this cell, how this cell will behave under this condition, and it will create the genes of the cell with that perturbation. And also conditional cell generation, when you can specify different type of conditions and you want to see the different genes of that cell. And also just natural language, where you ask, okay, how are tissues resident memory CD8 T cells adapted by the intestinal environment, and it be able to provide answers. So this is one of the core functionalities of the model that they developed and what they wanted to accomplish.
Pastor, can I just jump in here real quick? So just
Yeah.
for those who, for whom it's a little unclear, what the model is working on is RNA expression, meaning the genes in DNA are then translated to RNA, and RNA is then codes proteins, basically, to be very brief. And so like when you have this ranked list of genes here, gene T, gene A, gene S, whatever, those are a ranked list of how prevalent is the RNA that corresponds to that gene in the cell at the time it was measured. Just to be clear, and that's why there's this like sort of time dependent thing to it, where you have this, for example, the perturbation response is you have the expression when before the perturbation, and then you do some intervention like a drug or something, and then afterwards you have a different expression of genes. So it's the same, maybe exact same genome, but the expression levels have changed because of the environment. So I just want to be clear about what we're talking about. We're talking about RNA expression, not DNA.
Yeah. It's RNA expression, and yeah, thanks for that. And let me give you a brief of how cells behave and the cell cycle. And the cell cycle has different checkpoints. And basically, what the cell cycle does is that it has different checkpoints. And at any stage of those checkpoints, there are questions that the cell will pass or fail. And if it fails one of those checkpoints, the cell will kill itself from the inside. And for instance, in this checkpoint, it will ask for nutrients, growth factors, DNA damage. And if it pass at any of those levels, it will kill it from the inside. But if any of those checkpoints still fails, or that's what happened when you have a tumor cell, that these checkpoint don't work anymore. And for those situations, you have your immune system. So essentially, your immune system will detect those cells that that didn't pass that didn't have the checkpoints in place, and it will kill those cells. But cancer has already developed mechanisms like hiding some specific proteins that would allow your immune system to detect when these cells is behaving in or when these cells are have abnormalities. And on those cases is where the body develops cancer. So just a brief of introduction, like the cancer is a heterogeneous group of disease. And one thing that it has in common is that it is a proliferation of cells, and that didn't pass the checkpoints, but also the immune system is not able to kill it because it hides from the immune response. And that will become important when we see the methodology that they use. And yeah, so there are some type of medication that allows to detect those cell cancer, and we will see this specific treatment, how they tested and what they found using this model. So now we start the paper. The paper starts, the core functionality of the paper is to have the cells. You have the information of the cell, and you want to translate those information to sentence, and they wanted to use the LLMs to make sense of this biological data, and using LLMs to get insights into generative and also predictive capabilities in single cell RNA. And they use fine-tuning with reinforcement learning. One thing that they did is they also evaluate perturbation response, natural language interpretation, and complex biological reasoning that we will see in later in the discussion. And they also were able to validate and test in human cells, which was one of the most surprising stuff that I found in this paper. So basically, it's like getting a digital twin of a cell. They are able to graph the information of the cell and have in a virtual environment with the LLMs, and start to test different things with natural language model. So one of the accomplished is to have cell-to-sentence, which provides a framework to leverage the large language model for biology by transforming high-dimensional data into textual format. And data transformation strategy that simplifies the model development and deployment and enables easy integration on transcriptomic data with diverse modalities, including metadata, experimental conditions, and also textual description from biological publications. The accomplishment that they have is to have the C2 cell-to-sentence scale, which is a new family of large language model that was training on a multimodal corpus of over 15 million cells with associated text. And also they allow flexibility into the context to analyze cellular interactions and diverse biological information in multi-cell contexts. And that enables sophisticated application for predicting perturbation response to answer complex biological questions. And we will go into the details on the reinforcement learning, but they use GRPO to align predictions with key biological objectives, and they also propose a new metric for assessing generative performance, which is single cell threshold insertion distance. So this is an image that can summarize like the data set that they use and how they use it. Essentially, they have a vast amount of data, and that data is a multimodality data, which allows to have metadata, but not only for the cell itself, but also for research publications that you have from this cell and different behavior of the cell with perturbation, disease, and and a wide range of context diversity to try to have everything into context of that cell. And also they have different scale from gene, cluster, tissue, and donor. And what this does is that they allows to just ask questions about the data and even generate new data from the context. Are any questions so far? or clarifications, anything? Let me see if there is some.
I have a quick question.
Yeah.
I didn't read the paper, but I'm curious. So all this data they collected from their own experiments, or they're disparate data sets they've tried to join together. Basically, do they take the
Yeah.
same tissue test? Yeah.
So yeah, there is data that is already available, but what they did is that they collect data from different sources, and from those sources they aggregate the data, having we will go into the details of that, but essentially they aggregate data from different places, and but for different tasks they use different approaches.
Thanks.
Okay, so yeah, like I was mentioned, they use over 800 data sets, and the data sets have different biological annotations like cell type, disease, tissue, perturbation, species, cell state, and also they have metadata that has papers, and they use the abstract and this discussion and results from those papers to have their context. Also, they have gene sets and they have the interactions between genes. So essentially, what they did is that they have the you have the you have your you have the cell, and that cell has different genes, and those genes are ranked based on the activation and based on the expression that they have, and they rank based on that expression. So you have the genes that have the highest expression for that cell at the top of your rank, and then you started to you go in the certain order, essentially. So for instance, cell one has these genes, and these genes they have the highest expression in that specific cell, and they go in the certain order. So this allowed to have the fingerprint of the cell, but also allows to preserve the biological meaning of the gene expressions. So that's one of the clever ways that they use for actually preserve the information, and they did that with high accuracy.
So so this is a thing they didn't touch on the paper, but I think it's super important to understanding why this technique is really effective. Is that the gene, and I think they did themselves a disservice by just saying gene B, gene D, whatever, because I believe that the way South the sentence works is it has the actual sort of conventional annotation of the gene name. So like all these genes have names.
Yeah.
And that those genes occur throughout the popular, like the scientific literature. And that those genes appear in the context of, well, gene up, like gene, whatever is, like it's like known to be
Mhm.
prominent in like sort of breast cancer, like it's upregulated in breast cancer, and it like creates this protein, and it has part of this, sort of sort of this cellular mechanism, et cetera, et cetera, and all that is baked into the tokens that are in the gene name to some extent, and so that you're not just creating a sentence that is just a ranked list of arbitrary names, but you're actually doing a ranked list of things that have a lot of semantic meaning embedded in them.
Mhm.
So I think that's a really important point to understand about this.
Awesome. Yeah, thank you. And yeah, like they when they provide the metadata, they claim that because I didn't find a sample of the data with the actual metadata that links to the papers, they have some variation that will show in Embed. But yeah, like what you're saying is true. Like they also claim that they have they it's not only the gene. So they have a lot of information. It's the gene, then the gene name. It also has the gene expression by the rank, but also they link that gene to more information, which is the context that I'm providing here because it has the papers, every research paper that they have about that gene or about that cell as well, and the interaction between different genes and cells, which allows to have a wide context for actually making sense of the data that they have.
Yeah, and just to further emphasize the point that it's not only the context that they provide here and in the sort of fine tuning, but actually the base model has already been trained with all that knowledge, right? So they're building on it, and the fine tuning and reinforcement learning tells it how to respond, but it's actually kind of built into the base model as well.
Yeah, yeah, thanks. Okay, so one thing that they accomplish is cell type annotation, which is essentially passing the genes, the gene sentence or the cell sentence, and evaluate from which cell this belongs. So that's the cell type annotation, and they claim a 95.43% of accuracy in that specific task. Also, they have reached cell embeddings when giving a cell sentence as input. And so like a essentially is having the cell sentence and then transform it through different transformation into a numeric value, and then going from that numeric value to the actual gene expression. So they allow to have transcriptional and contextual information, and have a embedding with a lot of information, and they also support generative and interpretive in functions. So this is the chart that they use to describe the performance across different tasks. And as you can see, basically they beat every model that they test with this C2S scale. And in different tasks, this annotation of immune cells, this is perturbation response, question Q&A, interpretation held out studies that we will see later in more details, and captioning, also embedding with long cells, and also embedding with immune cells. So they essentially claim that they beat these models in every of these tasks. So let's try to go into the details on what this means, so we can kind of remove a bit of the abstraction that we have. So this is one demo example, and this is the prompt. The following is a cell sentence of a Homo sapiens cell. Your task is to give a cell type which these cells belong based on its gene expression profile, and they provide the cell sentence, and the output of the model is where these belong, or what's the type of cell. In this case, this is just an example, and you can see one thing that I found or that I think is more remarkable about this research is that they will allow to scale with the growth of LLMs. So as the LLMs became better, now that they have this approach to input the cells information as sentence, they will start to scale with the advance of the large language models. And you can see the different the BERT score across different trainable parameters. And this is dataset interpretation, for instance, and the following are cell sentences for different Homo sapiens cells. Your task is to produce an abstract summary summarizing the biological information of these cells, and they provide the cell sentence, and they get an abstract summary, which is what they say cell interpretation. But also they have generative capabilities, and they have generated cell sentence for 7 different Homo sapiens cells from a kidney tissue, and they provide a different cell sentence, or different variations of cell sentence based on what you provide here using all the contextual information that they have. And also you can generate cell sentence for the different Homo sapiens cell based on the following biological paper abstract and everything samples, and they generate different cell sentence for different variations of what is being asked here. And as you can see, that they test everything in the bear score, and they have training parameters. I think the most important thing from this finding is that they will scale with the large language models. Let me see if are there any questions so far? Yeah, I think that yeah.
I have a question.
Yeah.
Just wanted to confirm. So the input for this model is a sentence of genes all the time, or does it take in a gene expression vector?
Okay, we will see a table because for different tasks they have different inputs and different outputs, and I will show you a table that summarizes what inputs are needed for what task, so we can see it there.
Okay, awesome.
So,
thank you.
Let me just field that a little bit too. So they take the typical single cell data comes in the form of a cell by gene matrix, right? So you have
right.
And so what they do is they take the genes and they transform them basically into a ranked list of genes for a given cell, right? So you just order them by the expression level. You cut out the actual expression level and you just have a sentence.
Got it.
Does that make sense?
That makes sense.
And there's some plots
Yes.
in the paper about why this is like a reasonable thing to do.
Okay, yeah, thank you very much.
Yeah, thanks. So, okay, so the data collection, they have 50 million single-cell transcriptomic profiles from human and mouse tissue, and each dataset contains the arrow-provided metadata, which contains cell type and tissue annotations, donor information, and the processing pipeline goes through the regular processing pipeline, quality control, library size, normalization, and log transformation. And yeah, and this what the cell sentence is, they have each cell has the different range of genes, right? And those genes are ranked by their expression level, and they order those genes based on the expression level, which is what is providing by what this is. This is what is represented in this image. And this is what I was mentioning about the table. So they have different tasks, and these different tasks has different input information that they need, and they have a target output. For instance, this cell type annotation that we discussed, it has a single cell sentence that we saw as a representation that has gene A, gene B, gene C, so on, and they provide the target output, which is cell type. But there are other inputs. For instance, this conditional sample generation use a paper abstract, and they create multiple cell centers. That is kind of the example that we went when we were evaluating retinal cells. And so there are different inputs for a different task, and depending on the output that you need. Okay, so this is what we were discussing. What the gene A, gene B, gene C was a representation on what actually is, because there is no gene A, gene B, gene C. They provide the actual genes representation, and this is kind of what it looks like, what the data looks like. And they have the input representation. You have the cell name, you have the different sentence, and those sentences are ranked by the expression, by their expression, and then they are tokenized preserve the positional encoding to preserve gene rank order. So these also have that information. And the architecture that they use transformer, but only with the decoder, because they wanted to preserve sequential data and also support generative tasks. Now going through the pre-training stage, they use next token prediction. What they mean by next token prediction with this data is to predict the next gene in the rank order expression list. So based on the previous genes that you have, what's the probability to have the next gene? And that allows biological meaningful output for cell generation tasks and also data set level interpretation and Q&A. And okay, so how they evaluate, they use bear score for predictive tasks, and they use cell type annotation and data set interpretation, and they use gene overlap for generative tasks. Essentially, they compare the reference with a generated gene, and they evaluate the overlaps to see and they translate that to the bear score to have the evaluation. Okay, the post-training stage, they use supervised fine-tuning. They use the same objective as the pre-training, which is next token prediction. And they have the prompt, which is the cell sentence, and they have the prediction is the tissue of the origin of the cell. So as you can see, you have here the genes, and you have some metadata associated with that. You have the cell type, the tissue, batch condition, the organisms, and the cell. That's why I was mentioning that I didn't find a data that contains all the information that they that they that that they have with the papers, abstracts, and discussion and everything. But this gives a high level overview on how it works. So for the post-training part, they use GRPO, and let me briefly explain what this means and how this works. So you have a prompt. Let's say you have a prompt 12 times 30. You have that goes to a policy model, and that policy model generates different outputs, and those outputs goes to a reference model and to a reward model. The reference model, the goal of this is to not lose the gains that you gain through in the first part of the training. So it evaluates the child divergence and then go back to the policy model to keep to keep the outputs within the gains that they gain that they gain from the first part of the supervised fine tuning. And also you have a reward model that evaluates the different outputs and provides a score. And this is just a simplification on that reward. And from that reward, for instance, if you have the correct answer is +10, and if you have a wrong answer, -5, and you have a good answer, but it has more information that you need is +8 because it's not as a good answer as the first one. And then it goes to a group calculation, which is essentially a score to evaluate the mean and the standard deviation and evaluate the advantage, which is how much you gain relative to the overall rewards that you have in your space of rewards. And that goes back to the policy model, and they apply that to the token level. So they use for the post training, they use reinforcement learning alignment, and they use PBO and GRPO, which so are there any questions so far?
Maybe I missed it, but like when you have the tokenization, there's like a separate tokenizer that they have specifically for the genes, right? Or are they using
Yeah.
an off-the-shelf LLM tokenizer?
Mm, I'm not really sure. I didn't went that deep into the tokenizer. I think they use a different. I don't know if RJ wants to say
Yeah, no,
something.
they use the standard. So I think they were a little vague. I noted this in the thing. They think they just use off-the-shelf Pythia and off-the-shelf Gamma 2, and that includes the tokenizer. So that's sort of goes to the point that I was making at the beginning about how
Yeah.
it's actually advantageous to use the standard tokenizer, because then you pull in the knowledge about those specific genes as part of the um as as part of the token.
Okay, so
Yeah.
they're not actually trading from scratch, they're just fine-tuning Pythia and Gemma 2. Is that what you're suggesting?
So they were really vague about it in the paper, but I'm 90% sure that's the case.
Okay,
Yeah,
but even then, yeah, Pythia and Gemma 2, yeah, I guess, yeah, maybe that should be studied more, like the knowledge, because they're pretty small models, right? Like, I would be surprised if there is actually significant amount of like knowledge about these genes.
yeah,
I don't know.
unclear. I mean, but one thing that I noted from the chart in um a few pages before was that like it like the performance increased up to about 2B and then plateaued up to 27. And
Right.
so my interpretation of that was that there was the fine tuning actually played the biggest part there, and not the pre-existing knowledge. That's my
Okay.
like vague interpretation of that. Does that make sense?
Yeah, that makes sense. And then for the GRPO, yeah, so what exactly is the so the SFT, there, there were specific tasks, but for the GRPO, what exactly are the tasks that they're doing that on, I guess?
Yeah. So for the GRPO, they use that for the post training, and they use the advantage was calculated through the BERT score, and
Okay.
yeah.
I think that the table one that you had up above it might have the training type in it if it was like a few pages before. Yeah, here. Oh
These are
no,
all
I guess it doesn't.
pre-training.
Yeah, these are the tasks, but I can't remember which ones were GRPO. I think it was basically the ones that were
BERT
like
score. Yeah.
uh like uh like question answering ones, I think. But it's in the paper. I forget
Okay.
which ones. Yeah.
Mm. Okay, so let me explain the downstream task that they wanted to use. So the first one is the cell type annotation that we saw or that we discussed, in which you have the cell sentence and you ask for what cell this belongs, and also the cell cluster, which captures the surrounding of the cell and from where that cell belongs. And also they have the data set interpretation, which is with is asking at the tissue level and then asking to the organisms level. So they have a different range of tasks that they wanted to test. And this one, I found this one of the most interesting, because this is a special niche prediction, and what they try to do, they use location and context, or from the context getting the location of the cell. And as we mentioned earlier, this context is not only by the cell sentence, but also papers and research and gene interactions that they have. And for that, they use a human liver data set, and they cap down to the ratio of a neighbor to be 20 microns from the cell. And so this is what they try to do. They use niche level prediction, and this essentially is to give a cell sentence for a single cell and predict from where it belongs, or if the of if the cell belongs to a specific niche of cells, and also they have conditional neighbor generation, which is given multiple cell sentence from a neighbor, generate a null cell sentence that will belong to the same neighborhood. So we will see that in an example, but also they have a spatial neighborhood prediction, which is given multiple cell sentence. They predict whether the cells come from the same neighborhood, and the same niche prediction, which is given multiple cell sentence, predict whether all those cells have the same niche label or different niche. And let me show you what this means. And so you have, this is the first task. So the first task. So you have a cells with a cell sentence, and you wanted to predict the niche. So what the example that they provide here is that based on the information that we have about these cells, this belongs to a tumor niche. The other one is to having is to generate the neighborhood. So you have the cell sentence, and the generative task would be create the neighborhood of that cell. And the other one is the neighborhood prediction, and you have different cell sentence from different cells, and you wanted to ask if those cells belong to the same neighborhood, and also if the same niche prediction. So if you have a different range of cells, if they belongs to the same niche or not. And they evaluate different models on these generation capabilities. And the thing that I found surprising about this, or the application about this is not discussed in the paper. It's just my interpretation about what I think this can be really useful, is that when you have metastatic cancer, you sometimes you need to treat the primary tumor cells before treating the metastatic cancer. But that sometimes is not that easy. And the current methodology that doctors have for that is to analyze where that came from, and it's a pathologist that use research to try to identify where that belongs. But if you have the cell sequence of that tumor, you can predict where where's the niche of that cells, and from that started to develop a treatment. So I think for me it was a surprising thing, and a fascinating thing to see. Okay, now going to the Q&A. So for the dataset construction, they use GPT 4.1, 4.5, and they use abstract discussion and results, and they generate 20 Q&A pairs, and they use that for supervised fine-tuning. And from the supervised fine-tuning, they use the GRPO to generate 600 samples from unseen studies. And the candidate answers was 32, which is what I explained on the policy model, and the reward score was BioBear, or the advantage. So going through this, you have so this is how essentially it will work with the context. So the context say, okay, you have inhibitory neurons in the thalamus that can be identified by genes involved in the GABA synthesis and neurotransmission. So this is theory, these are facts that you have in your context. So you have the cell sentence, and with a gene ranking and everything, and you ask the question, is this given cell a inhibitory neuron? So the reasoning behind this, or the process, is that it will identify those genes and saying, yes, these two genes are present in cells that are from neurons that can produce GABA. So because you have those genes in that sentence, that came from a inhibitory cell. And as you can see, it has the GRPO with the policy model, and what I explained how the advantage was calculated. And this is the BioBear scores across different models. So yeah, like this allows to have like a virtual cell or a digital twin of a cell. And from that, a lot of research can be made. So now, like the core or why this paper was so discussed, or one of the claims of this paper that this research can provide, is that they actually test some hypotheses that they have, and they were able to identify interesting information. So as I mentioned, sometimes the tumor cells can hide from the immune system, so they don't attack those tumor cells, and then start to grow and develop cancer. So they test a treatment, or they test several treatments to identify and a specific hypothesis. This is one of the things that I wanted to highlight about this research, is that they don't get everything from the LLMs. They already have a hypothesis on what to test, and they actually was trying to find which drug was able to increase the expression of a protein that signals that is a cancer cell or is a tumor cell for your immune system to start to attack. They have a clear hypothesis that they wanted to test. So that's what they have. They did a virtual screening. So let me go through this. They test two different approaches. One is a immune context positive, which is a environment with low interferon levels, and they test which drugs increase or they test different drugs to see which one express more levels into that protein that I mentioned, that signals that you have a tumor cell. And they test in two different conditions. One with the immune context positive, which is low interferon levels, and the other one is in context neutral, which is with no interferon levels. So what they found is that actually there are some candidates, but the one that has the best results, which is this treatment, which is silmitacept, and they compare, as you can see here, like the immune context positive was able to evaluate that protein that signals cancer, or the expression trigger for the immune system to start attacking that cell. And like basically they count how many of these protein they have, and the research, they test without the treatment. They test the response without the immune cells response without the treatment, the immune cells response with the treatment, but in absence of interferon, and the immune cell response with treatment and interferon levels, and they see a remarkable difference between both. This is what I was mentioning. So you have here this on the x-axis you have the drug level, and on the here you have different type of interferon presence. You have 0 interferon presence, and you have 2 interferon presence, which is a low level, and this is the normalized evaluation of that protein that signals, or that signals that the immune response. And as you can see, there are clear difference, but those differences are not seen in only with the treatment. They are seen on the presence or absence of the interferon levels. So it was really specific about it, and this is what I was mentioned. This is low interferon level, and the response with this tumor cell was low, but the response with this tumor cell on the presence of low interferon level was high, or was a strong response to that specific protein. So the other thing that they test is perturbation prediction, and this they use data set from immune cells that exposed to individual and combinatory cytokines, and they test around 133 conditions where they evaluate cell type, stimulation, and exposure length. So this is an example data set that they provide in a different paper that they use. So you have the perturbation, the different type of perturbation that they evaluate. You have the cell type. So they test different combinations of this perturbation, and so for the fine tuning, and they use different levels of perturbations by by holding out some specific conditions to evaluate the generalization ability of the model. So the first one was cell type cytokine and only one condition, either chronic or acute condition on the perturbation level. And an example, B cells, the training that includes cell exposed to chronic interferon. But the test set required the model to predict the response to acute interferon beta. So the challenge it was to generalize in the time-dependent effect of the familiar perturbation. So they already have the perturbation, but it's the time condition that they wanted to test is it is able to generalize, even that it didn't see how it responds to that time of perturbation. The other one was a cytokine perturbation condition was excluded. And for example, you have a CD4 T cell where they held out for interferon gamma stimulation, and natural killer cells were held out of interleukin 6 stimulation. So the challenge here was the model to predict unique biological reaction of a known cell type to a specific but entirely unseen single cytokine stimulation. So essentially, you allow a perturbation, but they already know some information, but that specific one it doesn't have it haven't seen on the data, so it needs to generalize to this unseen scenario. And the last one, which is the hardest one, was that this. So it was testing a combination of two different perturbations, so that the model had never seen before. As when we saw the data set, we saw we have single perturbations, but also we have combination which between different cytokines. And now the challenge is for the model to predict combination of different perturbations that doesn't have. And as you can see, they started from the lowest complexity to the highest complexity and try to see if the model is able to generalize well or not. So one of the advantage of this research paper is that it allows to frame like a massive amount of context and information, and started to test hypotheses that without this tool or technology wasn't possible, because there is so much data that they have, and this is a fine-grained level, but we have data available to even find to even more detailed level. And the combination that you can test with drugs research paper, and the combination of different factors that change the environment where these cells perform and which these drugs can perform in the different conditions is just tremendous. So this technology allows to actually starting to make sense of this data. So the conclusions that I have for the paper is that the models are meant to test and evaluate theoretical assumptions by scientists and leverage their ability to reasoning on different approaches, and also understand cell language. Cell language provides a new space of possibility for research and treatment of cancer, in which genes plays a crucial role. And also LLMs specific to use case can outperform generalistic LLMs at a fraction of cost, which allows to grow and start to test more new things. So yeah, questions, comments, everything's welcome.
Hi, it's a very interesting paper. I was just wondering to what extent is this thing contaminated with the answers? So when you have an example that they had with a drug, a well-known cancer drug, I mean, somewhere it's probably going to say that this cancer drug is, you know, responsive to interferon gamma naught or whatever. It's somewhere in there. You could have just like, I just looked at that drug and I searched for it, and ChatGPT gave me like essentially a sentence saying it would be interesting. It's a good drug for cancer in this condition. So I wonder if they're just contaminating themselves with their kind of cell to text information. It's in there somewhere, right? I mean, you could potentially just run ChatGPT and ask. I mean, it's a really interesting system, right? But I
Yeah.
was wondering if you could comment on that.
Yeah. So when I was reading the paper, I have a similar concern because it has a lot of data and is using information from a lot of different places. And one thing that they tried to do is that they wanted to have, or because you have the gene level response of the cells based on those conditions, so you can kind of get the fingerprint of what changed based on the information that provides. So it was a really specific detail that low interferon levels actually help with this, or it says it has a synergistic effect into the drug and the interferon levels. And they also test in human cells, and then found the results replicable into what they found with the large language model on on the virtual screening that they did. So yeah, I think definitely it can be some type of contamination, but as you see, they try to allow different type of perturbation levels to see how good the model is able to generalize or not.
So
Yeah.
can I jump in here? Hi, Eugene. Eugene and I work together. There's a couple of charts that kind of address this. Let me see if I can. So first of all, let's see. There's two charts I think that are relevant here. This one here. Let me get rid of this. So this is not the same exact question, but this is in the question answering part, and they did compare against the baseline of LLMs so that their performance against after their fine tuning against these question answering prompts is like significantly better there. So that's one point. The other point that I thought was interesting was this one, which I think is more directly relevant, is that like so this is their model, and if you look at these t-SNEs or whatever they are, UMAPs, and you look at the ground truth of the perturbation pre and post perturbation classification, then they obviously are doing significantly better than these other single cell models, but it's a little unclear how much of the data is coming from just the base model versus the you know, sort of like insight that they got from the fine tuning. Does that make sense? I mean, they do show that these were things. Yeah. I guess it doesn't exactly get at your question, but I think it's relevant. So anyway, that's like what I wanted to add.
Okay.
Yeah. Cool.
Thanks.
Pastor.
Cool,
Thank you very
nicely
much.
timed. I think that is like roughly the chat, but yeah, I would love to have Tadish present at a future one, or like to start the for Science Pod. I think that's like, but no, thanks, Pastor, for volunteering. I think always actually very educational. I always feel like I'm attending school when doing this because of all the background needed, but very important for explaining all this. Cool. All right. I gotta go. I think we all do. Oh, Raphael, you're here. Hi. All right. Thanks, Sahil. Thanks, Tyler. Thanks, everyone. Thanks, RJ.
Thanks, guys. Bye.
Bye, everyone.